Hi, Miss Nikki here. Welcome to Chapter 2, Part 2. First, let's talk about organic molecules. So organic molecules are always going to have carbon and hydrogen. Carbons can bond with each other and form hydrocarbons. So you can see here we have the carbons lined up, and then you're always going to have, remember, 4 bonds. When we looked at that octet rule, we said in that first ring, carbon had 2. In the second ring, carbon had 4. So 4 electrons, it can hold 8, so it's got 4 spaces left. So you can see a carbon always likes to bond to 1, 2, 3, 4 other atoms. You'll also see carbon will sometimes double bond with itself, and I'll show you that when we talk about saturated and unsaturated fats. We do group organic molecules. So if we have these long chains of carbons, we'll look at the ends and you'll see some of these groups, these functional groups, like a hydroxyl group will always have an OH on the end of it. Carboxyl group will always have this carbon-oxygen double bond and then this hydroxyl group on the end here. So we group them based on their structures at the end, and this forms functional groups, meaning that whatever structure is hanging off of the end will tell you how that organic molecule is going to behave. So this should kind of remind you of, we talked about the outer ring of electrons on an atom, and that told you how the atom was going to behave. That's the same for these functional groups. Whatever's kind of hanging off the end is going to participate in reactions, and it'll tell you how that group of organic molecules is going to behave. This is a nice figure to show you how we can group together organic molecules or pull them apart. So 2 words you need to know. You need to know what a polymer is, and you need to know what a monomer is. So polymer, poly means many, mono means one. So if I have one monomer and another monomer, so I've got 2 different organic molecules and I want to join them together. Basically, you're going to remove water, you're going to do a dehydration reaction, and you're going to form a bond. And now the 2 monomers are joined, and so now we call that a polymer. The opposite reaction is shown here at the bottom, hydrolysis. I like it when they name things appropriately, right? Dehydration, remove water. Hydraulic break. So this is break with water. So if I have down here a polymer, right, we see the bond, I have a polymer and I want to split it. I do the hydrolysis reaction, I insert water, and now I have monomer 1 and monomer 2. So this is really important. I know it seems, silly. Okay, great. We put things together, we bring food in the body. So let's say you just ate a meal and you have a bunch of monomers. You have a bunch of glucoses. So you've got individual glucose. You've got glucose 1, you've got glucose 2, you've got glucose 3. So I've got these sugars, but I don't need them. I'm going to go to bed. I just ate dinner. I'm tired. I'm going to bed. So what can I do with those glucose molecules? I can do dehydration reaction. I can link all of the glucoses together, and I can store them as a polymer. I can store them for later use. You can store it in your liver. You can store it as fat. And let's say you get up the next morning and you say, oh, I feel terrible. I ate dinner and I went straight to bed. I just feel terrible. I'm going to go for a run. So you take the stored glucoses that you consumed the night before and stored, and you're going to do hydrolysis and break them down. Remember, you need individual glucose. You need 1 glucose to turn into about 36 ATPs. And ATP is the energy that you use to do work in the body. So the ability for us to do dehydration reaction and hydrolysis allows us to take in food and store it for later use to break down and get energy out of our food. If you didn't do this, if you couldn't do this, you would have to eat every single time you wanted to do some work in the body. Here are the categories of organic biomolecules. So bio, life, right? So these are the carbon and hydrogen containing molecules that are necessary for life. So we have carbohydrates, we have lipids, we have proteins, we have nucleic acids. So over here we have the polymers. And then over here we have the monomers. So if I take a monosaccharide like glucose and I link it with another monosaccharide, I get carbohydrates, which is the larger polymer. If I take fatty acid tails and link them together, I get a lipid. If I take amino acid 1 plus amino acid 2 plus amino acid 3, if I link amino acids together, I get a protein. If I link nucleotides together, I get a nucleic acid. So we'll talk more about these at the end. Here we have the structure of a monosaccharide. So remember, this is the monomer. We're going to take a bunch of these, we're going to link them together, and we're going to come up with our carb or our polymer. So these monosaccharides, they can have 5 carbons or they can have 6. So this one is 1, 2, 3, 4, 5, 6 carbons. The reason we talk about the 5 carbon is this is what makes up the sugar backbone. So DNA and RNA are made from a 5 carbon sugar, whereas carbohydrates are made from a 6 carbon sugar. So glucose is kind of the important, human body monosaccharide. This is the one that we're going to break down, basically store energy as ATP and use that energy to do work in the body. I wanted to talk about disaccharides next. It's when you join 2 monosaccharides. We haven't really gotten to big polymers yet. Those are coming up in just a second. But a disaccharide is just taking 2 sugars and linking them together. So we have maltose, which is if you take a glucose and a glucose and you link them together via the dehydration reaction, you get 2 saccharides, disaccharide, called maltose. Sucrose, this is table sugar. This is like the sugar that you put in your coffee. And then lactose is milk sugar. So these are relatively easy to break down unless you're lactose intolerant and you don't have the enzyme to do that. But for the most part, disaccharides are pretty easy to break down. Here we have polysaccharides. So this is more than just 2 sugars linked together. This could be hundreds of sugars linked together. So I would know this term, glycogen. This is what we store in our liver. So you know how I explained to you the dehydration reaction and hydrolysis? This is where we store that energy to use later. So we store excess glucose as glycogen in our liver. Plants create starches. So here she's got the potatoes. So this is a plant polysaccharide. It could be anything. Plants, wheat, carrots, veggies even have starches in them, right? So the plant was going to use this starch to grow. So the potato didn't make the starch for us. It made it so that the plant could grow again, right? And you can see that these look very similar. So there's not too much difference. This is why we can break down starches. We can pull out an individual glucose and we can use it to make ATP. This probably looks a little more complicated to you, and it should. This is cellulose. You see how there's hydrogen bonds forming here? Remember those dashed lines mean hydrogen bonds? You kind of form this sheet, and this is really hard to break down. So our bodies are not meant to break down cellulose. You might have heard of this as dietary fiber. So if you eat something high in fiber, it actually cannot be broken down. You can't get the nutrients out of it, but having fiber in your diet is good because it scrapes down the intestines and kind of cleans your system out. Now we move on to fats and oils, and this category is called lipid. So remember I said the fatty acid tails were the monomers, and if you link it all together, then you end up with this fat molecule. So glycerol is here at the head, and we call these fatty acid tails, and we link them all together, and now we have a fat molecule or a lipid. There are 2 different kinds of lipids. I just wanted to show you what a saturated fat looks like. So a saturated fat, do you see how there's no double bonds? It's just individual carbons, and then it's a whole bunch of hydrogens. So there's no double bonds with the carbon, and it forms linear fatty acid tails. So they're just straight tails. Saturated fats usually are solids. So here they give you a nice image of butter, right? And there are solids in milk, we just don't know because it's been homogenized. So this is anything that if you left it alone, it would take some sort of a solid form. Unsaturated fats mean that the carbons are not completely saturated with hydrogens. So instead of having hydrogens, sometimes carbon will share electrons with itself and form these carbon double bonds. And that allows for these kinks to happen in the tail. So remember the saturated, they kind of stacked up and formed solids. Well, in the unsaturated, these tails kind of stay wavy, and so you're always going to have a liquid. So why is it that unsaturated are good for you or better for you? Because they're liquid form, they're not going to get stuck inside your blood vessels, right? They're not going to form solid plaques inside your blood. So everything previous, we were talking about fats and oils, right? This is the second type of lipid, and it's got the name in it, lipid, right? This is a phospholipid. Remember, the fats and oils had 3 fatty acid tails. Oops, fatty acid tails, there we go. This one only has 2. This is a really, really important lipid. Without this lipid, you can't survive. So phospholipids make up the plasma membrane of cells. So here's the head, and here's the fatty acid tail, and here's the head, and here's the fatty acid tail. So it forms this, they call this a phospholipid bilayer. So 2 layers, one on the inside, one on the outside, and it separates the inside of the cell from the outside of the cell. So you could say if you had no fats in your diet, you would not be able to make enough phospholipids. It would not be good for you. So you've got to have some fat in your diet. This is the third type of lipid. So remember, we're still talking about lipids. This is the third category, and they look a little bit different. They have these 4 fused rings. So steroids, like testosterone and estrogen, they can direct meiosis, which is the development of egg and sperm, right? Cholesterol, most of us think of cholesterol as bad, but you do have some cholesterol wedged in the plasma membrane of cells. So just like we talked about the phospholipid bilayer, you're going to have some cholesterol in there as well to help give the cell membrane kind of some, they call it rigidity, they give it strength. So cholesterol is necessary. You need it to create the plasma membrane that wraps around all cells. Okay, so we've done 2. We've done carbs and we've done lipids. So we've got 2 of the biomolecules covered. We now move to the third biomolecule, which is a protein. Remember, proteins are made up of amino acids. So amino acids are the monomer, and protein is the polymer. So here are some common amino acids. They've given you the name and the 3-letter abbreviation. And again, these amino acids behave a certain way based on their structure. So depending on what group is hanging off of the amino acid will tell you how it's going to behave. And one of the things to notice here, do you see that there's always this nitrogen group hanging off? That's how you know that it's a protein. We need to talk about peptides, kind of another term for a protein, or it's the beginning of a protein. So if we have a peptide bond, that's going to join 2 amino acids. So you can see from this image, it's amino acid 1 plus amino acid 2. Look, we're doing dehydration reaction, right? We're removing water and we're joining the 2 amino acid monomers together, and they call this a peptide bond. So here we have a dipeptide. Well, guess what? If I added 1 more, if I added amino acid 3, this would now be a polypeptide. So these terms are kind of confusing. Again, think of the overarching umbrella is a protein. Polypeptide. peptides, which are proteins. They're not fully functioning proteins. Usually when we use the term polypeptide, we've just kind of joined the amino acids together. We haven't folded them and shaped them into functional proteins. So protein would be the overarching umbrella or big category. Polypeptides are inside there. They are proteins. They're just not fully formed proteins yet. And then what makes up a polypeptide? That's going to be amino acids. There are many different types of proteins. So we have structural proteins. Obviously, they give support. Collagen is found in our skin, right? It gives our skin the ability to bend and bend back, stretch skin. Keratin, hair, nails. Enzymes. These are proteins specific proteins that speed up chemical reactions. I call enzymes proteins that have a specific job. So we're going to see at some point an enzyme called sucrase that breaks down sucrose. And hopefully when you went back and looked at those, you saw that every time we have a sugar, it ends in ose. I'll tell you that every time you see an enzyme, it ends in ASE. And they're usually named after what they do. So sucrase is the enzyme that breaks down sucrose. Hormones are another type of protein, and these can send messages within the body. Hormones like we mentioned, testosterone, estrogen. Actin and myosin, these are both proteins that are inside your muscles and make your muscles contract. Then your muscles can't contract. That means your heart can't beat, your blood vessels can't pump, right? Your skeletal muscles can't contract and move you. Some proteins can transport molecules in the blood. So some proteins are going to grab a hold of a substance or a molecule, and they're going to move it from one area of the body to another. Antibodies that protect you, we think white blood cells, right? You may have heard of antibodies before. We have T cells and B cells. B cells are the ones that generate antibodies against a foreign pathogen. And then sometimes proteins can form channels. So you'd have your plasma membrane here, and you'd have a protein channel that allows substances to move from the outside of a cell to the inside of a cell. So you can see proteins have a lot of different jobs, right? It's not just one job, it's a multitude of jobs. So we do have levels of protein organization. So remember we were talking about individual amino acids linked together by peptide bonds, and we talked about forming polypeptides. So here's amino acid 1, 2, 3, 4, 5, 6. I'm not going to keep going, right? Link them together by peptide bonds and you form a polypeptide. Kind of think of a polypeptide as immature protein. Why? Because proteins have to be folded into specific shapes in order to carry out those various functions. So enzymes have to fold in a very specific shape in order for them to do their job. So if you disrupt the shape of sucrase that we just talked about, it cannot break down sucrose. So the shape is very important to the function of a protein. So I think of primary structure, this is kind of like beads on a string, right? We've just taken a bunch of amino acids and linked them together. We haven't folded anything yet. Secondary structure, you have two options. You can take this bead on a string and you can loop it into like this ribbon shape, and that's called an alpha helix. Or you can take the primary structure and you can kind of loop it and create this sheet-like structure. And so this is called a beta sheet. Now, third, tertiary structure, above. So what's in a tertiary structure? I could have primary beads on a string, then I could have secondary alpha, or I could have a secondary beta. So let's see if we can find this. Here's beads on a string, here's an alpha helix, and here's a beta sheet. Fourth, I can have a quaternary structure. In a quaternary structure, I'm taking a bunch of tertiaries and putting them together. So here's a tertiary, a tertiary, a tertiary, a tertiary, and now I've joined them together and we call it a quaternary structure. So this would be really large. We only have a few of these. Collagen is one of those examples. Collagen, let me finish spelling for you. So basically what we're saying is proteins have to fold in a very specific way in order for them to carry. So this slide is kind of just reaffirming what I said. The final shape of the protein is very important to its function. So the amino acid sequence is important. If I have 1, 2, and 3, I'm just giving you a number. They actually have names, right? But amino acid 1, 2, and 3, I'm going to get protein A. If I have amino acid 2, then 3, then 1, I get a totally different protein. I'm going to get protein X. So the amino acid sequence is important, but the final shape of the protein is also very important. If you lose the structure, you also lose the function. So proteins exposed to extreme heat or pH differences will denature. And probably the best example of this is an egg. So if you take an egg that was supposed to be a baby chicken, right? We took it away from mom, it didn't get fertilized. What happens if I fry that egg in a pan? Can I ever take the fried egg and turn it back into the egg and fertilize it and develop a baby chicken? No, never going to happen. We're finally on the fourth biomolecule, nucleic acids. So there's two types of nucleic acids. There's DNA or deoxyribonucleic acid, and then there's RNA, which is ribonucleic acid. So the structure is always a phosphate, a 5-ring sugar. Remember we said in the beginning there are some 5-ringed sugars. And then a nitrogen-containing base. So this is a nucleotide. If you remember, nucleotide was the monomer. So a phosphate, a sugar, and a base make up a nucleotide. That's the monomer. And if I have nucleotide 1 plus nucleotide 2 plus nucleotide 3, that's going to give me the polymer, which is called a nucleic acid. Let's see if I can squeeze it in here. Here are the similarities and differences between DNA and RNA. So DNA has a deoxyribose sugar. It's missing an oxygen. And RNA has a ribose sugar. DNA has bases adenine, guanine, thymine, cytosine. Some people say GCAT because they can remember that. And then this one has adenine, guanine, uracil. And this is important. So you're always going to see, or if you see uracil, it's always going to mean RNA. DNA likes to form double strands, and RNA forms single strands. DNA forms that twisted helix shape, and RNA does not. It's usually just single-stranded primary structure. Here's our structure of DNA. It forms this double helix. It's basically, if we pulled it apart, it would form a ladder, and it kind of looks like a twisted ladder. So you have bases in the middle. That would be kind of the steps of the ladder. And the sides of the ladder, you're going to have phosphate, sugar, phosphate, sugar. You're going to have alternating phosphates and sugars. The bases, again, are A, T, C, and G. So you can see here A and T bond together, and C and G. And you remember what those dashed lines mean? Hydrogen bonding, right? So one hydrogen bond is weak. Lots of hydrogen bonds are pretty strong. So we have C always binds with G, and A always binds with T. Here's a more detailed structure showing you the complementary base pairing. So remember, these are all nucleotides, right? And what's going to happen is they're going to link one base to another base. And remember, we'd have the sides of the ladder here, the phosphates and the sugars. So I know that this is DNA because I have thymine, right? So what would be here if it was RNA? You still have the C and G binding, and then you have A, and instead of T, you have U. ATP also falls under the umbrella of nucleic acid. Remember what a nucleotide consists of, right? A nucleotide is a sugar, a phosphate, and a nitrogenous base. So ATP is a sugar, a phosphate, and a nitrogenous base. But instead of linking them together to form a nucleic acid, it's just one. So this right here, ATP, is really a nucleotide, a sugar, a phosphate, and a base. What's interesting about ATP is that we can store energy in these phosphate bonds. So when we talk about cellular respiration, we're taking glucose, we're breaking the bonds, we're pulling out the energy, and we're storing that energy in the bonds of the phosphates and making this molecule called ATP. Now this ATP can be shuttled anywhere in the body, and what happens if I break this bond? I can release energy. So basically ATP is just an energy shuttle. It takes energy from one area in the body and drops it off at another area. So to kind of tie up the end of this chapter, I want you to realize that DNA or nucleic acids are linked to proteins. You can't have one without the other. So here we have our DNA with our nitrogenous bases. So there's a section of DNA, a gene, it's made up of these specific letters in a certain arrangement. I just made this one up off the top of my head, right? So this DNA is going to be transcribed into something called RNA, right? And then RNA is going to be translated into a protein. So basically, the RNA structure, this structure, is going to be translated into an amino acid sequence. And then that amino acid sequence is going to be put together, create a polypeptide, and create your protein. Then that protein has to be folded, right? So we get to that functional structure. So I just want you to realize that nucleic acids and proteins are connected, that you can't have one without the other. And we'll talk more about this as we move through cell structure and function.